So the next topic that we'll be talking about is epistasis. Epistasis is when one allele which is present in one specific locus of one specific gene is going to stop the phenotypic expression of another allele which is present in another gene in another locus. Okay, so you are going to think of two different genes. Let's think A and B. So there are two different genes which have two alleles, obviously. So you have a dominant allele A, a recessive allele B, a dominant allele B, and a recessive allele small b. Alright. So if you think of two different genes A and B, and then they have their alleles dominant and recessive, capital A and small a, capital B and small b for the different alleles of gene B. Now if you think that one of the allele of one of the gene which is present at a specific locus. So this one has a different locus than this one. So these two are present at two different locuses. Alright. So if one of the allele of one of the gene present at a different locus is going to stop the phenotypic expression of another allele of another gene which is present at another locus, then that process is called as epistasis. Okay. Now there are certain things, or rather certain terminologies that you will need to know with epistasis. One is epistatic gene. So what is an epistatic gene? An epistatic gene is the gene that is going to stop the phenotypic expression of another gene. So if you think back to the same example of A and B, let's say that the dominant A allele of the gene A is going to stop the phenotypic expression of the dominant allele B of the gene B. So this gene which is stopping the expression of gene B is called as epistatic gene. So the gene which is going to stop the expression is known as the epistatic gene. Another term is the hypostatic gene. So hypostatic gene is the gene that is getting stopped. Alright. So if A is going to stop the expression of B, then B is known as the hypostatic gene, because this one is getting its expression stopped by gene A. So gene A in this example would be the epistatic gene, gene B would be the hypostatic gene. Now there are two different kinds of epistasis, dominant epistasis and recessive epistasis. Now dominant epistasis is the case when the dominant allele of one gene is going to mask or stop everything else. Okay, so if you go back to the same example of gene A and gene B, so you have gene A, the dominant allele capital A, the recessive allele small a, and then you have gene B, dominant allele capital B, and recessive allele small b. Now imagine that gene A is going to mask the phenotypic expression of all the other alleles. So all the other alleles phenotypic expression, it's very much important that you remember this, that it's not the genotype that is getting changed, but rather the phenotype that is getting changed. So if gene A, which is the dominant allele of gene A, is going to mask or stop the phenotypic expression of all of the other alleles, then this kind of epistasis is known as dominant epistasis. Okay. Now, recessive epistasis is when, take the same example, you have capital A small a, capital B small b. Now, if you think that the gene B recessive allele, which is small b, is going to stop or mask the phenotypic expression of all of the other alleles. So, all of these alleles are going to get masked by this recessive allele of gene B, then this kind of epistasis is known as recessive epistasis. So, dominant epistasis when the dominant allele is doing the masking, and recessive epistasis when the recessive allele is doing the masking. Okay, I'll be explaining each of the epistasis, dominant and recessive, with examples. First off, dominant epistasis. Now, dominant epistasis can be explained taking the example of summer squash. Summer squash is a kind of vegetable belonging to the Cucurbita species. It comes in 3 different colors: white, yellow and green. Now, this is dominant epistasis, and white color is dominant over yellow and yellow is dominant over green. Now, this coloration is controlled by 2 different genes, A and B. Okay. Now, B gene is the one that is the dominant epistatic gene, meaning that when you have either homozygous dominant B or heterozygous dominant B gene, then the color will always be white. Okay. As long as you have the gene B in the dominant condition, meaning it is either homozygous dominant or heterozygous dominant, then the color of the summer squash will always be white. Only when the gene B is in a double recessive state, then it can give rise to either yellow or green. Okay. Now, we will understand it better when we do crossing. Okay, if you're going to do crossing, then you're going to take parents which are homozygous dominant and homozygous recessive. Now, if you're taking a homozygous dominant parent, because the gene B is in a homozygous dominant state, the color of this parent would be white. Now, if you are taking a homozygous recessive, because the gene B is in a double recessive state, it cannot be white, but also because A is in a double recessive state, the color is going to be green. So you are taking a white colored squash plant, crossing it with a green colored squash plant, and that is the parent generation. The F1 generation, of course, you are going to get a heterozygous dominant for both the genes. So you are going to get A with one dominant, one recessive, B with one dominant, one recessive. Now, because the B gene is in a heterozygous dominant state, the color of this one will also be white. So remember what I told you, that as long as the gene B is going to be in a dominant state, either homozygous or heterozygous, the color of the squash will always be white. Now, if you do F1 self-crossing, as in you're going to cross one F1 generation with the same F1 generation, okay, the usual Mendelian ratio or phenotypic ratio should be 9:3:3:1, right? This we have studied already when we were doing dihybrid cross in Mendelian genetics, that if you are taking two different characteristics and crossing them in the F2 generation, the phenotypic ratio will always be 9:3:3:1. But in case of this example of summer squash, the F2 generation had a ratio of 12:3:1, which is very different from the phenotypic ratio of the F2 generation in case of Mendelian genetics. So this is the ratio that you would get in case of Mendelian genetics. This is the ratio that you will get if it is dominant epistasis. All right. So going back, we know that whenever the B gene is in a homozygous dominant or heterozygous dominant condition, the color will be white. So what happens when the B gene is in a homozygous recessive condition? If it is in homozygous recessive condition, but the gene A is in homozygous dominant or heterozygous dominant. So B is in homozygous recessive, but A is homozygous dominant or heterozygous dominant, then you have the yellow color. All right. Now, if you have both the genes as homozygous recessive, meaning A is homozygous recessive, B is also homozygous recessive, then you have the color green. So the majority of them are white. There is a small minority which is yellow, and only one which is homozygous recessive, giving rise to green. This is how dominant epistasis looks. The only difference being that the F2 generation phenotypic ratio is going to be 12:3:1 and not the Mendelian ratio, which is 9:3:3:1. So this is with dominant epistasis. Okay. Going with the next kind of epistasis, which is recessive epistasis, I'll be explaining it with the example of coat color in mice. Now, coat color in mice comes in three different colors: the wild type or agouti, which is the black, then you have brown, and the white coat color or the albino. So there are three different coat colors in mice: black, brown and albino. Now the coat color are controlled by two different genes, C and D, and in this example C is the epistatic gene. Now because this is a recessive epistasis, only when the C gene is in a double recessive state does it become epistatic. Okay, so because this is recessive epistasis, the C gene needs to be in a double recessive state. If it is in homozygous dominant or heterozygous dominant, it is not going to mask. Only when it is homozygous recessive, it is going to be masking the other two colors, the other two coat colors. Okay, now to figure it out, we are going to do crossing again like we did with dominant epistasis. So we are going to take parents who are homozygous dominant and parents who are homozygous recessive. So homozygous dominant parent, okay, crossing it with a homozygous recessive parent. Now because the C gene here is in homozygous recessive, the coat color for this parent will be albino. Right? Like I explained before, because this is recessive epistasis, as long as gene C is going to be double recessive, homozygous recessive, it is going to mask the gene B, and therefore the coat color is going to be albino. Now what happens with gene B itself when the gene C is not homozygous recessive? So what will happen when you have gene C in either homozygous dominant or heterozygous dominant? Then gene B is the one that is going to decide whether it's going to be black or brown. So if gene B is going to be homozygous dominant or heterozygous dominant, the coat color is going to go to black. Of course, gene C will have to be homozygous dominant or heterozygous dominant also, because we know that if it is going to be homozygous recessive, it's going to become albino. Okay, these two will give rise to black. Homozygous dominant for B or heterozygous dominant for B in combination with homozygous dominant for C or heterozygous dominant for C. So this combination is going to give rise to black color. Now if you have the same homozygous dominant C or heterozygous dominant C with homozygous recessive B. So B is in a homozygous recessive form. This combination is going to give you brown color. Okay. Now if you are going with this, then here the homozygous dominant is of course going to have black color. So this parent is of black coat color. So this is the parental generation. It's going to have black coat color. And because the other one, which is homozygous recessive, is recessive for C gene, it is going to have albino coat color. Now, if you're going to have the F1 generation from this crossing, you're going to have a heterozygous dominant. So, you have C, which is heterozygous dominant, B, which is heterozygous dominant. Therefore, this color is going to be black again. Right? Now, if you are going to do a selfing of the F1 generation, so crossing the F1 with F1 is going to give you F2 generation. Now, Mendelian genetics will give you a F2 generation ratio of 9:3:3:1. But in case of recessive epistasis, you're going to have a F2 generation ratio which is 9:3:4. Okay? So, when all can you have black color? You will have black color as long as one of the B gene is in dominant condition and one or both of the C genes are in recessive condition. So the majority 9 will be dominant C or heterozygous C with homozygous B or heterozygous B. So this combination will be the dominant one which is going to give black color. Okay. Now where does the 3 come from? That is with brown when you have homozygous dominant C or heterozygous dominant C in combination with homozygous recessive B. So this combination is going to give you brown color, which is 3. Now albino is going to be when you have homozygous recessive C in combination with either homozygous dominant B or heterozygous dominant B or homozygous recessive B. So it can be homozygous dominant, heterozygous dominant or homozygous recessive. So any of this combination is going to give you albino. This one is brown, right? So in the epigeneration, you should, according to Mendelian genetics, have 9:3:3:1 ratio. But in recessive epistasis, you are going to have 9:3:4 ratio, in which case 9 refers to the majority, which is black, which is coming with homozygous dominant C or heterozygous dominant C in combination with homozygous dominant B or heterozygous dominant B. That is going to give you black coat color. Now for the number 3 here, you are going to have either homozygous dominant C or heterozygous dominant C in combination with homozygous recessive B, which is going to give the coat color brown. That refers to number 3. The other one is when you have homozygous recessive C. Like I said, this is recessive epistasis and C is the recessive epistatic gene, and therefore whenever C is in a double recessive homozygous recessive state, like here it is homozygous recessive in combination with homozygous dominant, heterozygous dominant or homozygous recessive B, is going to have albino, which is the number 4 here. The next thing that we will be talking about is transposons, or TEs as they are abbreviated, or they are also known as jumping genes. Now, like the name suggests, they are called as jumping genes because they basically jump from one location in the genome to another location in the genome. So they are going to move from one location in the genome to another location. This is not something that happens normally. So if you think of a gene in the genome, if it is present at a specific location, it always stays in that location throughout the generation in the entire population. So if you take human population as an example, and there is a gene for, let's say, eye color, and it is present at a specific location in the genome, it is going to be found there irrespective of whichever individual you pick up and test. But that is not true for transposons. Transposons are jumping genes, and therefore they are not going to stay in one location. So if they are, and they keep moving all through the genome. Okay, there are different methods by which they move, and that is how they are classified, but we will get to that later. So first things first, transposons are genes that are going to move from one location in the genome to another location in the genome. Now, because they keep moving from one location to another, they either create or reverse mutations. So because they keep jumping from one location to another, they either create a mutation or they reverse a mutation. Now, transposons was something that was discovered by Barbara McClintock in 1940s when she was working with maize. Alright. Now 50% of the human genome is made up of transposons. So 50% of the bulk of the human genome is made up of these jumping genes, which are going to keep jumping from one location to another location. In case of maize, it's even more higher. 90% of the maize genome is made up of transposons. Like I was saying, based on the way that they are going to move from one location to another, they are divided into two different kinds. One is class 1 transposons, which are known as retrotransposons. The other is the class 2 transposons, which is known as the DNA transposons. Now we are going to talk about each of these things individually and figure out what is the mechanism for each of them. First things first, we have the class 1 transposons, which is the retrotransposons. Now retrotransposons move from one location to another location through the process of reverse transcription. Now reverse transcription is a process by which an RNA can be converted to a DNA. Now you know that RNA and DNA are two different kinds of nucleic acids, and they are different based on the kind of ribose sugar that is present in them, and also the thiamine nucleotide which is there in DNA as opposed to the uracil which is there in RNA. Now RNA gets converted to DNA by the enzyme reverse transcriptase, and this process is called as reverse transcription. Now this is the process that retrotransposons use to move from one location to another. How? I'm going to explain now. Now imagine that there is a retrotransposon, and this is there in the genome of that organism. Right? It is present on the genome of the organism. Now this retrotransposon is going to get transcribed to RNA with the help of RNA polymerase 2. So with the help of RNA polymerase 2, the transposon which is there on the genome, so this is in the form of DNA, is going to get converted, or rather transcribed to RNA by the enzyme RNA polymerase 2. Now once RNA is synthesized, this is going to get converted into DNA by reverse transcriptase. So reverse transcriptase is going to convert RNA into DNA, and once this DNA has been formed, this is going to be moved to the target sequence in the genome. Okay, so you have a retrotransposon which is there in the genome at a specific location. This is in the form of DNA because it's there in the genome. Now this is going to get transcribed to RNA by the enzyme RNA polymerase 2. So now we have RNA which is there in the cytoplasm. Now RNA is going to get converted to DNA by the enzyme reverse transcriptase, and now this free DNA segment which is there is going to get integrated into a specific target sequence in the genome, and this space, this place where it is going to get integrated is very different from the place where it was before. So it is not going to get integrated at the same place, but rather it is going to get integrated in a very different place in the genome. So this is how retrotransposons move from one location to another. You have the transposable element in the DNA form in the genome, gets converted to RNA, gets converted to DNA, and then gets integrated in another location in the genome. So the next thing we'll be talking about is class 2 transposons, which is the DNA transposon. Now there are no active DNA transposons in the human genome, and therefore they are known as fossils. It means that the DNA transposons, they are there in the genome, in the human genome they are there, just that they are not transposing anymore, meaning that they are not moving from one location to another, they are just there stagnant. So they are not active, and therefore they are known as fossils. Alright, just the way you would refer to any archaeological find, this is also known as fossils, because they are not active anymore. Now DNA transposons are going to move by a cut and paste method. They are going to use a cut and paste method, and for the cutting and pasting they are going to use an enzyme transposase, and this enzyme is a endonuclease. Okay, so DNA transposons are going to move through a cut and paste method, meaning that they are going to get cut out of the genome from the place that they were in, and they are going to get pasted in another place in the genome. And the way the cutting and pasting is done is with the help of the enzyme transposase, which is an endonuclease. Now, endonucleases are enzymes which are going to cut the DNA at a specific location. They're going to make a cut in one location in one of the strands, and then in another location in another strand at the same kind of DNA sequence. So they're not going to cut anywhere and everywhere. They need a specific DNA sequence where they're going to cut it. Now, to understand it better, here is the genome, and in the genome you have the enzyme transposase gene. So this is the gene which is going to give rise to the enzyme transposase. Now, next to the gene on both sides are sequences which are known as inverted repeats. Or IRs. So on both sides of the gene you have sequence which are inverted repeats. So this is the gene which is going to give rise to the enzyme transposase. So the transposase gene is here, and flanking it on either side are inverted repeats or IR regions. Okay. Now what happens is that the transposase gene is going to get transcribed. So through transcription you are going to get the mRNA, and through translation you are going to get the protein. Now this protein is going to be nothing but the transposase enzyme. So this protein that you are getting at the end of translation is the transposase enzyme, and like I said, it is an endonuclease. So what it is going to do is to cut the genome at different locations, so that this part of the genome having the inverted repeats on both sides and the transposase gene in the middle is going to be removed out of the genome, and then it is going to be pasted somewhere else in the genome. Okay, this is the mechanism of it using the endonuclease enzyme transposase. Okay, there are two different kinds of DNA transposons: replicative DNA transposon and non-replicative DNA transposons. So there are two different DNA transposons, which is replicative and non-replicative. The next topic that we will be talking about is polygenic inheritance, or it is also known as quantitative inheritance. Now, from polygenic, it's a Greek term, when poly means many and genitus meaning to produce. So by the name itself, it means that one phenotypic trait is being controlled by many genes. So usually you have one gene. So if you think of tallness in garden pea, it is going to be controlled by the gene T, where it has two alleles, one dominant allele T and one recessive allele small t. Whenever dominant allele is present, the phenotype is going to be tall. Whenever recessive allele is present, the phenotype is going to be dwarf. This is with Mendelian genetics, the classical Mendelian genetics that we know. But in case of polygenic inheritance, it's going to be not one, but multiple genes that are going to give rise to one phenotype. So many different genes are going to give rise to one phenotype, and that is why it is known as polygenic inheritance. And also, these different genes need not be present on the same pair of homologous chromosomes. They can be present on different pairs of homologous chromosomes, but they are always present at different locus. So a phenotype which is controlled by more than two genes and each of these genes are present at a different locus, and they can be present on the same pair of homologous chromosomes, or they can be present on a different pair of homologous chromosomes, then this kind of inheritance is known as polygenic inheritance. Now, it is also known as quantitative inheritance because it depends on the additive effect of every single allele of every single gene. So if we think that there are 3 different genes which are going to give rise to one specific phenotype, let's for example think that there is a gene X, a gene Y and a gene Z. All 3 of them are going to give rise to one phenotype. Okay. Now each of these genes are going to have 2 alleles, obviously. So it is going to have capital X, small x, Y is going to have capital Y, small y, Z is going to have capital Z, small z. Now it's a quantitative inheritance, meaning that depending on how many dominant genes are present or how many recessive genes are present, it is going to give rise to different phenotypes, meaning this gene need not have a heterozygous dominant allele, it can be homozygous dominant. Okay, and let's say that this gene Y is going to be homozygous recessive and this gene Z is going to be heterozygous dominant. Okay, so you have 3 different kinds of alleles here, one being homozygous dominant, one being homozygous recessive and one being heterozygous dominant. It is a quantitative type of inheritance because you are not going to see which gene is in the dominant state, which gene is in the recessive state. Rather, you are going to see how many are in the dominant and how many are in the recessive. So you have 1 dominant, 2 dominant and 3 dominant. So because you have 3 dominant genes here, it is going to give rise to a phenotype which will be different from the kind of phenotype you would have in, let's say, if X was double recessive, Y was double recessive and only Z was heterozygous dominant. So you have only 1 gene which is dominant here. So the kind of phenotype that you will see with 3 dominant gene would be very different from the kind of phenotype that you would see with 1 dominant gene. So it is a cumulative effect and a quantitative effect. So it depends on the number of dominant or recessive genes which are present, not which gene is dominant and which gene is recessive. That's why it's also known as quantitative inheritance. The examples of polygenic inheritance are quite a lot. So one of the examples is height in humans, and this was studied by Sir Francis Galton. Skin color in humans is another example, and this was studied by C.B. Davenport. Intelligence in humans is also another example which was studied by Alfred Binet. Differential fertility in humans, kernel color in wheat, cob length in maize, fruit size in tomato, all of these are different examples of polygenic inheritance or quantitative inheritance. Now another thing to know is that, like in case of epistasis or like in case of any other non-Mendelian inheritance patterns that you have seen until now, the F2 generation ratio is going to be different when you think of polygenic inheritance. So if there are 2 genes which are involved in the production of a specific phenotype, then the F2 generation ratio, so if there are 2 genes which are involved in the production of one specific phenotype, then the F2 generation ratio is going to be 1:4:6:4:1. Okay, so normally if there are 2 genes which are involved, in case of, let's say, dihybrid cross, the F2 generation ratio is going to be 9:3:3:1. But in case of polygenic inheritance, if there are two genes which are involved, the F2 generation ratio is going to be 1:4:6:4:1. Now if there are three genes that are involved, then the F2 generation ratio is going to be 1:6:15:20:15:6:1. Okay, so if there are two genes involved, the F2 will be 1:4:6:4:1. If there are three genes which are involved, then the F2 is going to be 1:6:15:20:15:16:6:1. Sorry. So 1:6:15:20:15:16:6:1. So that is how the F2 generation phenotypic ratio is going to change from the classical Mendelian F2 phenotypic ratio. We are going to learn more about polygenic inheritance using skin color in humans as an example. Now skin color in humans is controlled by three different genes, A, B and C. And all three of them are present on three different homologous pairs of chromosomes. So you have one pair of chromosome on which gene A is present, another pair of chromosomes on which B is present, another pair of chromosomes on which gene C is present. Now because they are homologous pairs, there is a locus at which one of the allele is present. On the same locus on the homologous pair of chromosome, its alternating allele will be present. So the alleles which will be found here can be dominant A and A, or heterozygous in this way, or homozygous recessive in this way. Okay, and these can differ as in it can be A and A, or A and A, as in here it can have a dominant A allele and here recessive A allele, or here it can be a recessive A allele and a dominant A allele. But these are a homologous pair of chromosomes, and therefore they have a specific locus at which both of the alleles are present. And like I said, all the three genes are present on three different homologous pairs of chromosomes. Now because this is quantitative inheritance, depending on the number of dominant alleles present, if more dominant alleles are present, then the skin color is going to become darker. If more recessive alleles are present, the skin color is going to become lighter. So in that sense, if you have a homozygous triple recessive, so you have A which is triple dominant. So if you have A which is going to be dominant, homozygous dominant, B which is going to be homozygous dominant, and C also is going to be homozygous dominant, then the skin color is going to be the darkest skin color that you can find. Now the opposite of it, where all three of the genes A, B and C are going to be homozygous recessive. So both of the alleles of A are recessive, both the alleles of B are recessive, both the alleles of C are recessive, then in this case you have the lightest skin color. Now if you have a mix and match of all of this, in the sense that you have one dominant, one recessive for each, in this sense, so you have a dominant A, a recessive A, dominant B, a recessive B, a dominant C and a recessive C, then you are going to have a intermediate skin color. If you have a homozygous dominant for all of the genes, then you are having a darkest skin color. If you have a homozygous recessive for all of the genes, then you have the lightest skin color. If you are going to have a mix and match, as in you have three genes which are dominant and three genes which are recessive, then you are going to have an intermediate skin color. Now we can just plot it here as a reference point for us. So because there are 6 genes, you're going to because there are 3 genes, you're going to have 6 alleles. So if all 6 alleles are dominant, like here, then you're going to have a darkest skin color. 3 of them are dominant, you're going to have an intermediate skin color. So you have 3, then it is intermediate, then none of them are dominant, like in the double recessive, then it is going to be the lightest skin color. Okay, we can for ourselves give names for the darkest, lightest and the intermediate. Let's say that the darkest skin color is going to be black, wherein all 6 alleles are homozygous dominant. If there are 5 of them which are dominant, then you are going to have, let's say, a dark brown. If there are 4, then you have a brown. 3, you're going to have, let's say, a light brown. 2, you're going to have fair. 1, you're going to have very fair. And 0 is going to be the albino. Okay, so this is a graph that we are going to put here as a reference point for us when we are going to do the different kinds of meat. You are going to take a parental generation which is going to be homozygous dominant. So it's a capital A, capital A, capital B, small c, capital C, and then you are going to mate it with a homozygous recessive, so small a, small b and small c. Now, like I was saying, this would give the darkest skin color. So this parent generation is having a black skin color, and then you're going to cross it with a homozygous recessive. So this is albino skin color. Remember that this is the parent generation. From here you're going to get the F1. F1 generation is going to be heterozygous dominant for all the 3 genes. So you're going to have a capital A, capital B, small b, capital C, small c. Now, because it is having 3 dominant genes and 3 recessive genes, this is going to be a intermediate skin color. All right. Now, for F2 generation, you obviously are going to cross F1 with F1 again. Okay, so the gametes that you are going to get out of this will be 8, because you have 3 different heterozygous genes. So the first gamete you have is all dominant. The next one will be with 1 recessive. The next one will be with 2 recessive. The next one is going to be with 1 recessive and 2 dominant in this way. After that you have 2 recessive, 1 dominant, 2 recessive, 1 dominant, and all recessive. So you have 1, 2, 3, 4, 5, 6, 7, and 1 got missed, which was A, B and C. Okay, so you have 8 gametes which are forming here. Now, as a pro tip, if you want to figure out how many gametes need to form from a specific genotype, there is a very simple calculation that you can do, which is 2 to the power of n, where n is going to give you the heterogeneous gametes, that heterogeneous genes that are there. So, for example, let's say that there are 4 genes A, B, C and D. Okay, and out of these 4 genes, 3 of them are heterozygous and 1 is homozygous. So let's say you have a genotype which is in this type. So out of this, if you want to figure out how many gametes need to form, you just have to think of how many of them are in the heterozygous condition, which is 1, 2 and 3. All 3 of them are in the heterozygous condition. The D is a homozygous dominant. So you have to just insert N for how many of them are in the heterozygous condition, which is going to be 3. So 2 to the power of 3 is going to give you 8. So you are going to have 8 gametes being formed from this. So that's a pro tip that you can use whenever you want to make sure that the amount of gametes that you are having is the same. Now from these gametes you are going to do a crossing. So you are going to have a 64 Punnett square cross, which was we were trying to do the F2 generation cross from the F1 generation self crossing. So this is what you get from the F1 generation self cross. So this is the F2 generation where you're crossing all the 8 gametes with the 8 gametes here. This is a 64 square Punnett square where you're crossing all the 8 gametes with all the 8 gametes which you got from the F1 self crossing. Now from this there is something that you will realize which is very important for polygenic inheritance. It is that it does not matter which gene is dominant and which gene is recessive. What matters is how many genes are dominant and how many genes are recessive, because this is quantitative inheritance. So polygenic inheritance is quantitative inheritance, and therefore you are going to figure out how many of the genes are dominant and how many are recessive, and based on that the phenotype is going to change. Now before I went into this, I already wrote a sort of inference point here, wherein if you have 6 dominant alleles, then the skin color is going to be black. If you have 5 dominant alleles, it's going to be dark brown. 4 you're going to have brown. 3 you're going to have light brown. 2 you're going to have fair. 1 you're going to have very fair. If it is 0 dominant alleles, you're going to have albino skin color, which is devoid of any kind of pigment. So melanin production is not there in case of albino. Okay, once you do the F2 generation, what you are going to figure out next is how many dominant genes are present in each of the offspring. So starting off from here, you can see that there is only one offspring which has all 6 dominant. So all through the Punnett square, you can sit down and check for yourself. None of them have all 6 dominant. So there is only one which is going to have all 6 dominant genes, and because of that, that individual is going to have the darkest skin color, which is black, and from there comes the 1 here. All right, next is how many of them have dark brown, which comes from 5 dominant alleles. So 5 dominant alleles, you can check here, 1, 2, 3, not there, 4, 5, not there, and then not there, not there, 6. All right, so you have 6 offsprings which have 5 dominant alleles, and the dominant alleles are different in each of them. So here you have A, which is heterozygous dominant, so you have 1 dominant allele in A, 2 dominant alleles in B, and 2 dominant alleles in C. But if you take, for example, this one, you have 2 dominant alleles in A, 1 dominant allele in B, and 2 dominant alleles in C. So this is very different from this genotypically, but the number of dominant alleles are the same in both. So because this is quantitative inheritance, we are not going to consider which of the genes has the dominant allele, but rather how many genes have dominant allele. So anywhere there are 5 dominant alleles, does not matter which gene, but 5 in total, those offsprings are going to have brown color. So you have 6 of them, and there you get the ratio 6. The next one. Now the next thing is how many of them have 4 dominant alleles. You can count all through the Punnett square and you will find 15 of them. 15 of the offsprings have 4 dominant alleles. For example, here you have 4 dominant alleles, 2 in A, 1 in B, 1 in C. Again, you have 4 dominant alleles here, 1 in A, 2 in B, and 1 in C. Now this is very different from this, and like I said when I was explaining 5 dominant alleles, it does not matter which gene has it, but how many dominant alleles are present. So if you calculate, add up how many 4 dominant allele containing offsprings are there, you will find that there will be 15 of them. That is the next ratio that you get for the F2 generation. Next comes 3, and it goes the same. So you have 3 dominant alleles here, 3 dominant alleles here, 3 dominant alleles here. Again, very different from each other. Here you have 1 dominant allele in A and 2 dominant alleles in C, which is very different from here where you have 1 dominant allele in each. So there is 1 dominant A, 1 dominant B, 1 dominant C. Again, count the number of 3 dominant allele containing offsprings and you will get 20. So 20 of them will have light brown skin color. Moving on, 2 dominant alleles. Search through the entire Punnett square and you will have 2 dominant allele containing offsprings. Now how many of them will be there? 15. 15 offsprings will be there which will be containing 2 dominant alleles. And like every other example, here you have 1 in A, 1 in B, but in this case you have 1 in A and 1 in C, which are very different from each other genotypically, but what matters is how many dominant alleles are present, and because both of them have 2 dominant alleles, both of them will have the same skin color, which will be fair. And how many of them are there in the 64? There will be 15. Same goes for 1, which is very fair. You have 1 here, 1 here, and 1 here. Alright, so you have 1 dominant allele in A, 1 dominant allele in C, and 1 dominant allele in B. And same like 1 dominant allele B here, 1 dominant allele C here, and above that 1 dominant allele A. So if you count all through the Punnett square, you will find 6 of them which have 1 dominant allele. And last comes the last number 1, which will be all recessive or 0 dominant alleles. So there is only 1 offspring out of 64 which will have 0 dominant alleles, where all of them are recessive, and that offspring is going to have the albino skin color, and there comes the ratio 1. So the ratio 1 refers to black, then the next ratio second refers to dark brown, next ratio 15 refers to brown, which contains 4 dominant alleles. Then the next ratio 20, the biggest one, has the intermediate skin color, which will be light brown or wheatish you can call, which will have 3 dominant alleles. The next ratio 15 refers to fair, which has 2 dominant alleles. And then you have 6, the next ratio, which will be very fair, which will have 1 dominant allele. And the last ratio 1 comes from 0 dominant alleles, where all of them are recessive, which gives the albino skin color. So this is the example for skin color, which is a polygenic or quantitative inheritance, and this is the Punnett square, which is drawn from selfing F1 generation. So F1 generation is going to have a heterozygous dominant, where each gene has 1 dominant allele in it. If you're going to self it, you're going to end up with 8 gametes and 64 offsprings, out of which you're going to get all the different kinds of skin color ranging from black to albino. Okay. The next topic that we are going to talk about is the chromosomal theory of inheritance. Now, why chromosomal theory of inheritance? Because until now we have been studying Mendelian genetics and variations of Mendelian genetics, as in things like codominance or incomplete dominance or polygenic inheritance, wherein it is not following the rules of Mendelian genetics, but at the same time we are still talking about alleles, we are still talking about factors, we are still talking about genotypes and phenotypes, correct? So until now we have always talked about alleles and factors, as Mendel called them, and each factor had two different alleles. So if you're going to think of tallness as a characteristic, as a phenotype, now that factor is going to have two alleles, the capital T, which is the dominant allele for tallness, and the small t, which is the recessive allele for tallness, which gives rise to dwarfness actually. So if you have homozygous T or heterozygous T, you're going to have a plant which is tall. If you have homozygous recessive, then you're going to have a plant which is dwarf. Now this is one of the rules of Mendel, that these factors are made of two units, and each unit is going to segregate when there is gamete formation that is happening. So if you have a parent which is made up of genotype capital T and small t, when this parent goes for gamete formation, you are going to have capital T in one gamete and small t in another gamete. So this is where segregation is happening. So first we have factors. Factors have two unit factors, and both the unit factors are going to give rise to two different phenotypes. The next thing is that when you have a heterozygous organism, the factors are going to segregate into two different gametes when the organism is going to go for gamete formation. Okay, so this is something that we already know, already have studied all through Mendelian genetics and variants of Mendelian genetics like incomplete dominance or codominance or polygenic inheritance or epistasis or anything else that we have studied until now. We know that this thing works. The problem came when Mendel did not check what is happening at the cytological level. So what Mendel did was mostly breeding experiments. He was taking plants and then crossing them and figuring out what the offspring was, and based on that he was inferring what might be happening. But in the 1900s, microscope was discovered, and because of that people started looking what is happening at the cytological level, or rather at the cellular level. So to figure out how these factors are behaving at the cytological level, and that is when the chromosomal theory of inheritance came about. Now the chromosomal theory of inheritance was studied by two different scientists independently, so they did not work together, they studied it independently, but they came to the same conclusion. The two scientists who studied were Walter S. Sutton in 1902, and he was working on grasshopper, and the other scientist was Theodore Boveri, who was studying in 1903, and his study was done on sea urchin. Now what they were trying to figure out was how these factors are changing, or how these factors are being inherited through the process of reproduction. So for the process of reproduction, they were studying meiosis, because we know that gamete formation happens in meiosis. Mitosis is when one mother cell is going to give rise to two daughter cells. But in case of meiosis, you are going to have one mother cell which is going to give rise to four daughter cells. So that's the difference between mitosis and meiosis. And mitosis happens in the body generally when a cell needs to be replaced, when a cell is growing old. So one mother cell is going to divide and give rise to two new daughter cells. But meiosis is a process that is going to happen only during gamete formation. Okay, so chromosomal theory of inheritance is based on these two scientists' independent study on two different organisms when they both came to the same conclusion, and both of them were studying gamete formation and therefore were studying meiosis. The chromosomal theory of inheritance is also called as the Boveri-Sutton or Sutton-Boveri theory. Okay, and the experiment that they did can be explained in this way. So first you take a mother cell which is going to go through meiosis. Now you know that meiosis is going to take place in two stages. In the first stage, the mother cell is going to duplicate its genome during the interphase. So during the interphase, you have duplication of the genome. Okay, so this is the mother cell that you are going to take, and at interphase the genome is going to get duplicated. So imagine that the mother cell has, for this experiment, the example that I am giving, we are going to take example of two pairs of chromosomes and they are carrying two genes. So one pair of chromosome is carrying gene A and another pair of chromosome is carrying the gene B. Now the pair of chromosome which is carrying the gene A is going to have at one locus a dominant allele and at another locus a recessive allele. And the same goes for the pair of chromosome which is carrying the gene B. At one locus you are going to have dominant allele capital B, and at another locus you are going to have the recessive allele small b. So two pairs of chromosomes, each pair is carrying one gene A and B, and the pair is going to have two alleles having a dominant and a recessive, dominant and a recessive. So the pair which is carrying the dominant is shaded in this way, the crossed one, and the chromosomal pair which is carrying the recessive allele is not shaded. So just keep in mind all through the experiment, the shaded one is going to have the dominant allele for both, and the non-shaded one is going to have the recessive allele. Also, the pair which is carrying the gene A is going to be slightly bigger in comparison to the one that is carrying the gene B. So just keep that in mind all through the experiment. So this is the mother cell. We have two pairs of chromosomes carrying two different genes, and during interphase it is going to duplicate the genome. So when the duplication of genome is going to happen, you are going to have two copies of each of them. So you are going to have two copies of chromosomes containing dominant allele A, two copies of chromosome containing the recessive allele A, two copies of chromosome containing the dominant allele B, and two copies of chromosomes containing the dominant allele B. So after interphase, after the duplication of the genome, this cell is going to go through meiosis 1. So you have meiosis 1 occurring here. So from the mother cell you are going to get two daughter cells. So during daughter cell formation, the genome, the chromosomes are going to get distributed between the two daughter cells. So for example, let's just assume that all of the chromosomes containing the dominant allele for both the genes are going to end up in one daughter cell. So both dominant A's and both dominant B are going to end up in one daughter cell. The other daughter cell is going to have all the recessive allele containing chromosomes. So both the recessive allele containing chromosome B, both the recessive allele containing chromosome A are going to end up in another daughter cell. So after meiosis 1, these cells are going to go through meiosis 2. Now, the major difference between meiosis 1 and meiosis 2 is that before cell division occurring here, the cell is going to go through duplication of the genome. So, you are going to have, instead of 4, 8. and from there you get 4 again. So you are going to have the same chromosome number as that of the mother. So if you are going to think of this as a human cell, you are starting off with 46 chromosomes, correct, which is 22 pairs of autosomes plus the 2 sex chromosomes, which can be XX or XY. Now during the duplication process, you are going to end up with double the number of chromosomes. So instead of 46, you are going to have 92. So 92 chromosomes you have after interphase when the duplication of the genome has happened. Once division takes place, each of these cells are again going to have 46 chromosomes like the parent cell, like the mother cell. The daughter cells are going to have 46 chromosomes. Now when these daughter cells are going to go through meiosis II, they will not have a duplication step. So these daughter cells are going to divide without the genome duplicating, without the genome replicating. So the number of chromosomes are going to remain the same, 46, and this cell is going to go for division. So when this cell is going to go through meiosis and go and divide, each of these cells are going to give rise to 2 daughter cells. So on the whole, you have 4 daughter cells arising from one mother cell. Now each of these daughter cells are going to have 1 pair of chromosomes, right? So from the dominant A, you're going to get 1 here. From the dominant B, you're going to get 1 here in one of the daughter cells. Other dominant A and B are going to come to another daughter cell. Same goes for this, wherein 1 from the recessive A and 1 from the recessive B is going to come to one of the daughter cells, and 1 from the recessive A here and 1 from the recessive B here is going to come to another daughter cell. So because you are not going through replication from 46, each of these cells are going to have 23 chromosomes. So this is how gamete formation is going to happen. And you need to have 23 chromosomes because each of these gamete is going to get fertilized by another gamete. Imagining that all of these gametes are eggs, they are going to get fertilized by the sperm. Now because they have 23 chromosomes, the sperm will also have 23 chromosomes. When they come together and fertilize, the zygote, which is going to form after fertilization, will have 46 pair of chromosomes, which is the normal pair. That is the reason why gamete formation is going to occur in this way. Because if you ended up with another replication process here during meiosis II, each of these cells would end up with 46 chromosomes, and after fertilization the cell would end up with 92 chromosomes, which is not normal. That is the reason why this process needs to take place without a duplication of the genome. So now that this is done, we know that each of the daughter cell is going to have one of the allele from one of the gene. So if we take gene A, then one dominant allele is going to go to one daughter cell, one recessive allele is going to go to another daughter cell. So if you go back to Mendelian genetics and think that each phenotype is governed by one genotype which he named as factor. So instead of thinking it as factor, let's say that it is a gene. So for chromosomal theory of inheritance, the factor that Mendel said was actually the gene. Alright, now gene comes in pairs. What Mendel said was factors comes in pairs. Unit factors are there. So instead of unit factors, here you have alleles. Allele A, allele A. Okay, and after that, when you come to gamete formation, there is segregation of the factors that takes place in case of Mendelian genetics. The same is happening if you think of it as chromosomes. So chromosomes are also segregating into all the 4 different daughter cells. So chromosomal theory of inheritance is nothing but a physical proof, a biological proof at a cytological level, at a cellular level, that whatever Mendel postulated is true. That chromosomes carry the genes which he postulated as factors are genes, and they are carried on chromosomes, and the chromosomes are passed on from the parent to the offspring through the gamete, and at the gamete formation process is when the chromosomes are segregated, and therefore each of the allele is going to get integrated into a different gamete. And there you have the segregation taking place. So this is how chromosomal theory of inheritance is a physical proof at a cellular level, at a cytological level, for all of the theories that Mendel postulated.